Welcome to class. Today in Unit 4, Molecular Genetics, we're going to be focusing on mutations and what could go wrong in the processes we've discussed so far for protein synthesis. Just to recap, we've discussed DNA to mRNA, which is transcription, to get the message out of the nucleus. And then we've talked about mRNA to the amino acid polypeptide sequence, which is going to be the eventual protein when it's done, and that's translation, translating it from the nucleic acid language to the amino acid language. If there's an error in some of the coding in that process, that's what we're going to be focusing on, are the types of errors and what could result from those. So these are all about mutations. The big idea for the unit, once again, nucleic acids store directions to build and run organisms through the processes of transcription and translation. I've added the phrase using ribosomes because if we tied that organelle in specifically in the last lesson. Errors that affect the process, or I should say errors in the process affect the resulting proteins. And we're going to learn all about the specifics of that. In order to reach that big idea, I want you to focus on the learning target. Can you analyze a DNA molecule before and after a mutation and determine or predict which of 6 different errors was involved in the process of protein synthesis? 3 DNA mutations that we're going to focus on are deletion, point mutation, and insertion. 3 resulting protein errors we're going to look at are silent, missense, and nonsense. Now there are other errors and types of mutations, but we're only going to be naming these 6 and referencing them in this lesson. We predominantly have focused on this because of the cancer epidemic that we face as a people. And this illustration at the top just shows you how cancer can spread quickly. If you remember, during mitosis, a body cell, when it divides, is exactly the same. So if there's a mutation in this cell, when it divides, there will be 2 cells with a mutation. And then every time that DNA replicates, another set of cells will have the new mutation, the new coded sequence. And oftentimes those can be malignant or bad for your health. There are 3 categories of external sources for mutation. Before I cover those, I just should point out that mutation that happens is most often random. There are mistakes that are made in the coding processes of transcription and translation. Sometimes, to come back down to this illustration, there are external factors, radiation, chemicals, or infectious agents. So radiation from the sun or too many x-rays, not your normal amount of x-rays. Chemicals like cigarettes, processed foods, or some cleaning products that aren't regulated as well. And then infectious agents is probably not one you consider often, but a virus, for example, cannot replicate itself. It has to insert its DNA into its host organism's DNA sequence, and then whenever that organism replicates its DNA, it replicates the virus DNA as well. And there's also some bacteria that do that. So since we've learned that a gene on a chromosome, or a collection of nucleic acids on a DNA molecule, code for a protein eventually, what if there's a mistake on the gene or in the sequence of nucleic acids? This is called a mutation. Any change to the normal DNA sequence we would consider a mutation. So I am going to ask, if there's a change to the DNA, will there always be a change to the protein that is made? I want you to think about that for a minute. I'm going to ask you again at the end of the recording, and I want you to chime in. The first thing to discuss is the 3 types of DNA mutations that we're going to name in this lesson: insertion, deletion, or point. Sometimes we call it substitution mutations. I'm going to go into each of these in detail in a little bit, but the name of the DNA mutations are insertion, deletion, or point or substitution mutations. And they're going to be referring, those terms refer to the sequence of nucleic acids that are on the original DNA strand. You know this is DNA by looking at it because there is the presence of thymine. So this is not RNA, this is DNA. So the first one is insertion. If you look at the original sequence, you should be able to see that instead of reading TAA, CTG, the new sequence is TAA, CTG. So this cytosine nucleic acid has been added in by accident. It's as if it's squeezed in there. During the process of replicating the original DNA strand, there was an error and an extra nucleic acid was added. This type of DNA mutation ends up resulting in a shift in the reading frame. Remember, we call those reading frames codons. So if you're reading them in threes, if you insert another nucleic acid here, you're shifting the reading frame by one nucleic acid. We're going to call that a frame shift mutation as a result. Likewise, a deletion mutation would also result in a frame shift error in the protein. In this case, the original sequence would be TAA, CTG, etc. Instead, it reads TAA, CGC. So this codon is now altered. And instead of reading this frame, there's a shift in the frame. So you're reading it an error into the resulting protein that we're going to call a frame shift error. So insertion, deletion, these two, just to reiterate, result in a frame shift mutation in the protein. It changes every amino acid in the protein after the mutation. It shifts the bases over and messes up every other codon. So if the original sequence read CAG, CCC, ACT, which would get glutamine, proline, and threonine is the amino acid sequence, the frame shift mutation is going to read GACTCC, CACT. So at this point, there's an extra nucleic acid. This would have been an insertion mutation because you have an extra or several extra nucleic acids that you didn't have before. Well, what does that look like in terms of regular grammar? Think about it. If it were a deletion mutation and the sentence were to read, the fat cat ate the rat. If you take a C out, then it doesn't make any sense. The fat atta tat her ought, right? And so you've shifted if you have to read everything in words that are 3 letters long, you've shifted the frame of reading. If you insert one, instead of the fat cat ate the rat, it's the fat car tat asked era t. So just keeping that in mind, you're shifting the frame that you're reading. The third DNA mutation, we've covered insertion and deletion. The third one is point, sometimes called substitution mutations, where you're not adding or deleting. It's just a random error of what nucleic acid was supposed to have gone there. So during replication, the original sequence, TAA, CTG, somewhere in the replication process, it now reads TAA, CCG as the codon. But you're still reading it with the same number of codons in the sequence. Your frame of reading has not shifted. It does result in a typo in the reading frame, but not a shift. So when you're reading a passage and you come up with a spelling error or a grammatical error, sometimes it's nonsensical, right? Or a sensical error. So when we talk about that, there are 3 possible results in the protein from a point mutation. There is a silent error, there is a missense, and there is a nonsense. Those are the three types of errors in the protein. So, for example, these only happen with point mutations. We're not talking about insertion or deletion. We're only talking about after a point mutation, a silent error would not have any effect on the protein sequence. So, yes, there's a change from the original sequence, but the error still results in valine. So remember on our DNA codon chart, or I should say the RNA codon chart, there can be more than one codon that codes for the same amino acid. So a silent error would be that type of situation where instead of GTA, you have GTT, but both GUA and the mRNA sequence and GUU on the mRNA sequence both code for valine. So yes, there's an error, but it's silent. You don't notice it in the protein formation. A missense would be when it doesn't make any sense. You come up with an error that it doesn't read correctly. So instead of, let's see, AGC, GTA. So instead of CCC in this codon, that would eventually convert to proline. Instead of proline, you end up getting threonine. So the amino acid sequence doesn't make sense. It's a missense. If you were reading this sentence, you'd say, what is that word there for, right? That's a missense or a mistake, okay? Nonsense mistake or error, a nonsense error is very specific. A nonsense, I actually want you to think of it as a no sense, because no is going to mean stop. Instead of coding for tyrosine, UAC, the error mutation in the DNA strand, ends up, instead of coding for UAC, codes for UAG. And UAG is a specific codon for stop. So this protein is going to end up stunted. It's going to end up being maybe much shorter than it's supposed to. So it is a no sense. Okay, it is not a sentence. As if I were to say, the fat cat, and then I didn't finish the sentence about it eating the rat. It's just a fragment of a sentence. It's not a sentence. So silent, there's an error, but it doesn't affect the protein sequence. Missense, there's an error and it makes a mistake when you're reading the sentence. And then nonsense or no sense means instead of a word, you get a stop. So it's a fragment of a sentence. Here's an example that we can work through together. I want you to pause the video and try to answer this on your own and then check your answers. AA here means amino acid. So the normal DNA codon would be TTC. The new codon is TTT. So you'd have to find your mRNA sequence, which for us would be AAG or AAA for the mutation. Originally, AAG would be lysine, AAG, lysine, but now it's AAA. Oh, so that's still lysine, AAA. So a silent mutation is still going to create the same codon or the same amino acid. It's still going to code for the same because AAG, AAG, and AAA, AAA are both going to result in lysine. So all good. Missense is when a mutation causes one amino acid to be changed. So instead of TTC, you end up with TGC. Pause the video and tell me what's going to happen. So what we see is that the mRNA sequence should read AAG, but now reads ACG. AAG, G is lysine, ACG, A, C, and any of those is threonine. That's a different protein altogether because the sequence has a mistake. Nonsense. Okay, so a nonsense would be instead of coding for lysine, you code for the stop codon. Because UAG is specific to stop. Okay, then let's just look again at sickle cell anemia, only because it's a common enough genetic error that we can analyze it. We should have, at this point, be able to identify that sickle cell anemia is a point mutation. Sometimes it's called a substitution. But what ended up happening in the mutated DNA, that instead of the codon CTC, it was CAC, and the mistake is that valine is the amino acid that matches the mRNA codon for GUG. And instead of glutamic acid, you get valine. And that's what causes one little change in a nucleic acid sequence causes an entire change in the red blood cell structure. And again, those cause a lot of health problems. Now we're going to practice this tomorrow in class. So I'm not going to have you, I do want you to take notes. At this point in the semester, I've shown you how to take notes. I've shown you some examples of what note taking can look like. You're going to need to be able to do this throughout these kinds of lessons. So bring your notes to class when you come and we can practice this, okay? Take a minute and consider how you're feeling about the one learning target we've got to focus on, where you analyze a DNA molecule before and after a mutation and determine or predict which of 6 mutation errors was involved. So tomorrow we're going to practice going from DNA to protein and looking at that, and then the protein sequence backward and looking at that. So that's all I have. I will see you in class. Come prepared to practice and have a great day.